Perfect. Good morning. Welcome to the Blake's Breakfast Speaking Series on neurological diseases, and thank you for joining us today. My name is Ryan Butt, Director of Partnerships and Events. All attendees have their video off and are on mute. However, we encourage you to be active and participate in this event by posting questions in the Q&A section, where you can also upvote on all questions and use the chat functions as well. After each of the speakers have presented, they will be going into the chat and to the Q&A and can answer any of your questions. As we begin, I'd like to acknowledge that I am joining this event within the ancestral, traditional, and unceded territory of the Coast Salish people, including the territories of the Musqueam, Squamish, and the Tsleil-Waututh First Nations. I would also like to acknowledge that others are joining us from other traditional and unceded Indigenous territories across this beautiful province and country. We are excited to be here to host this event virtually. Over the past year, Life Sciences BC and our community have fortunately embraced virtual events. We are grateful that we can still continue to discuss and draw attention to the extraordinary work being done in the life sciences sector in BC. For those of you that do not know, Life Sciences BC is a not-for-profit society. Our members consist of academic and research institutions and local and global companies operating in the life sciences space. Example, digital health, medtech, med devices and diagnostics, and therapeutics, and those companies that provide support and services to the broad life sciences ecosystem. We are funded through membership and generous contributions of our sponsors and partners. This event was made possible by our event supporter, Blake's. We greatly thank you for your investment in LSBC and the life sciences ecosystem, which is invaluable, and we are thankful for your support. At each event, we like to announce our newest members. Today, we would like to welcome Azor Biotech, Gandiva Therapeutics, and Group Parima. We're always welcoming new members, and please reach out to any of us at LSBC or go to our website, lifesciencesbc.ca, for more information. So now on to the program. So I'd now like to introduce Kyle Miswich. Kyle is a partner at Blake, Castles & Graydon, LLP, practicing corporate and securities law, including public and private company financings, mergers and acquisitions, takeover bids, and corporate reorganizations, with a particular focus on life sciences and technology industries. He also advises clients in connection with securities law compliance, disclosure obligations, stock exchange matters, corporate governance, and general corporate law. Kyle is a longtime and passionate supporter of the life sciences sector. Over to you, Kyle, to introduce our first speaker.
Great, thanks Ryan, and welcome everyone. Blake's has been a long time sponsor of the LSBC Blake's Breakfast Series, and I'm looking forward to another great event. So I would like to introduce our first speaker, Simon. Dr. Simon Pimstone is a founder, director, and executive chair at Xenon Pharmaceuticals, one of Canada's leading publicly traded biotechnology companies. Xenon is engaged in discovering and developing novel therapeuticals targeting neurological diseases with a key focus on ion channels. Dr. Pimstone received his MD from the University of Cape Town. He is an internal medicine specialist. Prior to his specialization, he trained as a clinical research fellow with the Department of Medical Genetics at the University of British Columbia and obtained his PhD in cardiovascular genetics through the University of Amsterdam. Dr. Pimstone also serves as a consultant physician at the UBC Medical and Cardiology Clinic at UBC Hospital in Vancouver. Welcome, Simon, over to you.
Thanks so much, Kyle, and thank you, Ryan. Really great to be part of this breakfast series that you've sponsored for decades now, and a big gratitude that all of you have kept this community so close. So great to be here, and thanks for having me. I want to talk a little bit today about Xenon, but with a particular focus on neuroscience and neurology, which is really the theme today. I was asked just to present a brief overview of our core activities, and it's been a very busy few months for the company, so timing is obviously great to do this, but also perhaps just to leave with some themes that we certainly are seeing and thinking about in neurology more broadly, either as an operator or potential investor or a collaborator in this industry. I think neuroscience is really taking off in a very significant way today, and I like to think about this as sort of the next decade of the brain, and really catching up to where investment in oncology has been, and there are a variety of reasons for that. So we can discuss that later. So a bit about Xenon. We are a neurology-focused company. We set out very early in our establishment to focus the business in a core area of therapeutic area in neuroscience, but also in a core target class area being ion channels. And I'd say today, the company has probably built the premier ion channel discovery engine in the industry today. We've been at it a long time. As many of you know, Xenon's been around now, it's almost 2 decades, and we've had ups and downs, as many biotechs have. We've weathered the storms. And I'd say part of the reason we've been able to do that is really the focus, but the breadth in activities, and today have a very exciting pipeline, including multiple mid to late stage clinical development studies. And we believe we're a few years off from being a commercial company, which today I think is a really great outcome for a company that starts with a concept to be able to move all the way through the non-clinical and clinical realm into commercial. I think is a super achievement, and we're excited to be where we are, and as a company very proud to have gone through what we've gone through and hopefully come out a stronger entity at the end of the day. We're in a very good financial position with this. At the end of the last quarter, we had about 250 million in cash. We raised another 324 subsequent to that. So really the company is, as they say in the business, well cashed up, and we've removed really any source of financing overhang and have multiple years of cash on hand to do what we need to do. So the breadth and depth of the company is really shown on this slide. I'm not going to go through every one of these programs or products today, but just to give you a sense, we do have a number of products in late stage phase 3, our Xenon 496 orphan epilepsy program targeting kids with a condition called KCNQ epilepsy, a rare genetic form of epilepsy. The big news for the company in the last month or two has been around our product Xenon 1101, another potassium channel opener. We had very exciting phase 2 data recently in focal epilepsy. We've initiated a first major depressive trial with this, with collaborators in Mount Sinai, and we expect to launch our phase 2 sponsored study in major depressive disorder in 2022. We've got another number of ion channel modulator programs in non-clinical, and then a number of partnerships, which has also been an important part of the company's strategy and enabled good non-dilutive financing over the years with some very meaningful partnerships. Again, these are some in the ion channel space with Neurogen and with Flexion. I wanted to touch on perhaps the recent news from the company. It was really an exciting time for Xenon. As many of you have been in this industry for decades know, it really is at the end of the day all about data. It's easier said than done, as we know. There are a lot of trials and tribulations to get there, and I would say all of us have experienced more failures than successes in biotech. I think this was an incredible outcome for the company that really proved staying power, persistence, and perseverance can pay off, and we really had an amazing couple of months since the announcement of our top-line data from our Xtol trial. Xtol was a large phase 2b study in adults with focal epilepsy. This is a very interesting commercial opportunity. People may not be aware there are a couple of blockbuster products in this indication. A drug known as lacosamide or Vimpat, and another one known as Keppra. Really significant products in the epilepsy space. And so despite that, 40% of patients with focal epilepsy remain refractory with ongoing seizures. We had developed 1101 as a potassium channel opening drug, really exquisite non-clinical data to support its activity in animal models, as well as a human phase 1 study, which I'm not going to show you today, where we actually were able to show in a model known as a TMS, transcranial magnetic stimulation model, where we were able to apply a magnet to a human's brain. We were able to show the effect of this drug in excitatory pathways, both cortical and corticospinal pathways, giving us a very good indication in a phase 1 setting of the activity of the drug 1101 in the human brain. And I'll come back to that in my ending slide, because the importance of doing some of these quote unquote biomarker type studies, in this case, really a physiologic assessment of the effect of the drug on excitatory pathways, which is what you're trying to impact with an epilepsy drug. We were able to show that in a phase 1 setting, gave us a lot of confidence on the activity of the drug in the CNS, but also allowed us to dose select for the clinical trial. And so this study was a 8-week maintenance study. You can see the design here, 3 active arms with a 25, a 20, and a 10 milligram dose. Again, mid-dose set by our transcranial magnetic stimulation phase 1 study with a placebo arm. Patients were then either followed up or went into an optional open label extension. We have many, many patients, a very, very high conversion rate, over 95% of those completing went into the active arm in the open label and remain on that. And a number of patients have now completed quite extensive periods in the OLE. The data that we presented about a month or so back is shown here is really quite exceptional in a number of ways. The primary endpoint shown here is the change from baseline in seizure frequency, the median seizure frequency, comparing at the end of the study from baseline. And you could see a quite remarkable dose-dependent effect here, which is exactly what one wants to see in a clinical study. You can see clinical significance and statistical significance being met in the low-dose arm, but highly statistically significant at the 20 milligram and 25 milligram arm compared to placebo. So it's a very, very important slide for a few reasons. Number one is placebo has been changing quite significantly over the last decade or two. Many studies have shown very high placebo rates in epilepsy, as well as, of course, other indications. And the relatively low placebo rate we were able to achieve here, I think, speaks volumes to the conduct of the clinical study. We can go into this in much more detail. But how one actually sets up the study and the ways in which the study is conducted, I think, play a very, very important role in your placebo response rate. And we were able to keep that rate low. But what you can see at the top dose at about 53% median seizure frequency reduction is really as good as it gets in epilepsy. And I would actually say better than it gets. The blockbuster products that I spoke about earlier, generally those effect sizes are somewhere between our 10 and 20 milligram dose, around 30 to 40% seizure frequency reduction with drugs like Vimpat and Keppra for focal epilepsy. And we can see both our mid and high dose at significantly higher frequency reduction in seizures. So this was a very, very exciting outcome. Clear drug effect, clear drug dosage, very high numbers of patients, a large study of over 330 subjects, constituting a phase 2B trial with statistical significance being met across all doses tested. Of course, similarly, we were very interested in secondary endpoints, and in particular, the global impression scores in epilepsy, which are really a reflection of how the caregivers and how the patients see their response rates. And typically, these are looked at as and I'm looking now at the table on the right, the proportion of patients much improved or very improved. You can see, looking just, I've shown the top dose here versus placebo, a more than doubling of improvement on the 25 milligram arm in both how the caregiver global impression turned out, as well as the patient global impression at the bottom. And you can see on the left-hand side, this is now another key secondary endpoint. These are essentially response rates, the proportion of patients getting a greater than 50% response rate, which is deemed highly clinically significant. And you again can see a very, very nice dose-dependent effect with high statistical significance in the mid and higher dose, but meeting statistical significance even at the low dose. So very, very exciting clinical data from this trial, really, I'd say about best in class that's ever been seen in an epilepsy trial with an epilepsy product. The treatment emergent AEs in the double-blind period safety population data were also very encouraging. You can see this shown by placebo and then increasing doses as you go right in the table with any dose in the final column. And you can see the placebo and 10 milligram arm very, very similar in treatment emergent AEs as well as in serious AEs. And actually very exciting for us was no difference in serious AEs across any dose group. Some treatment discontinuation difference as you go higher in dosage, which is really not unexpected. And we had absolutely no serious AEs leading to death in the study in any group. So I would say, based on this data, a very, very exciting, group of data set, which at least speaks to a very significant benefit to risk ratio. So how we start to think about the clinical positioning of this product, obviously there are a number of drugs. This is a genericized market. Two of the blockbusters are shown in the top, levetiracetam and lacosamide. But these will generally be used in the easy to control patients. As I mentioned earlier, about 40% of patients with focal epilepsy, of which there are about 500,000 to 600,000 of these patients in the US, are refractory or difficult to control. And we really believe 1101 will be positioned as first in line in a rational polypharmacy to treat these patients with no concerning drug-drug interaction liability with this product either. So as we start to think about the product, and this is something I'd encourage companies to always think about, is it's not just about efficacy. Of course, the efficacy characteristics are shown on the left. We had very compelling data, very strong combination data supporting polypharmacy, as over half of the patients in the study were on 3 anti-seizure meds or thereabouts or more, but at least 3. Of course, we also may have a mood benefit. I'll go into that in a minute. But the other characteristics that really are going to drive the commercial opportunity here would be the ease of use and safety tolerability. This is a 1 pill once a day, no titrations required. It's a unique mechanism. There are no other drugs in the space that work in this way, and a very forgiving PK, an extensive terminal half-life that allows coverage when patients miss doses. And that's very important in epilepsy, where these patients have breakthrough seizures. Safety tolerability I've talked about. We have some other interesting work being initiated now in major depressive disorder for this drug. I'm not going to go into this in a lot of detail, but shown on the bottom is a precursor drug. We have this precursor drug in a phase 3 trial in a pediatric condition. But the drug was tested in a small, in fact, 2 small studies in major depressive disorder, a drug known as ezogabine. It's also a potassium channel opener. Our drug 1101 is a significant improvement on ezogabine. But you can see in both the MADRS scale shown on the bottom left, as well as the SHAPS score, which is a score of anhedonia, the green bar showing below the orange bar is essentially an improvement in both the anhedonic scale and on the left in the MADRS scale, an important marker of major depressive mood. And so we have some really compelling early clinical data with a drug, a predecessor drug known as ezogabine, which showed improvements in both major depressive scores and anhedonic scores in 2 clinical trials. We now, on the basis of that, have moved this product 1101 into 2 studies of major depressive disorder. I'm going to just touch on our partnerships. I mentioned earlier, these have also been a bedrock of the strategy of the company. We have a mixed model of both partnered and retained assets, and happy to go into reasons why we choose partners on certain products and retain others. But Neurogen has been a great partner on a drug we used to call 901. It's a sodium channel inhibiting drug. It's a very selective drug on the NaV1.6 channel. They're developing this drug in 2 clinical trials. We have those underway, both focal epilepsy as well as patients with a rare genetic form of epilepsy known as SCN8A. And then our Flexion partnership is with a drug we used to call 402, a NaV1.7 inhibitor, which is currently in a phase 1b bunionectomy trial for pain and should read out early in 2022. So one of the exciting things, as I started with earlier in my talk, was just really the breadth of the pipeline and the number of catalysts and value-creating milestones we have. 1101, obviously an important source of this value as we go forward. We've just had a successful phase 2b Xtol trial readout. We expect the end of phase 2 meeting with the FDA in Q2 of next year to support our phase 3 program in adults with focal epilepsy, and we're evaluating other epilepsy indications for 1101. 1101 is also moved into a major depressive disorder program, obviously a huge opportunity. There's exciting, both non-clinical, I never shared with you today, but also clinical data with a predecessor drug that acts in a similar way. So we're working with Mount Sinai on a phase 2 that's now started, and we have expect to start our sponsored phase 2 study in the first half of next year. 496, which is ezogabine, is in a phase 3 trial in kids with KCNQ, developmental epilepsy. It's a rare orphan disease, very, very severe form of epilepsy with major developmental impairment. And then partnered programs, which allow for some very significant milestones, both with Neurogen and with Flexion Therapeutics. I was asked just to end off with some key themes. One of the things we've really started to think a lot about in neuroscience drug development are non-invasive surrogate markers. Our human TMS, transcranial magnetic stimulation study, which essentially is a study that allows you to assess the effect of a drug on cortical and corticospinal excitation, was incredibly important for us to A, show we have drug effect in the brain, and B, to determine at what level or what dosage that effect was occurring. It allowed us to really select the dose for our phase 2 trial, and I think played a major role in the success of the phase 2 trial that I've just presented. So this, for us, is going to be a very important element of dose prediction and trial design, is to try and implement these non-invasive surrogate markers early in a phase 1 program. I think the other element you'll see a lot more of in neuroscience is the development of drugs in genetically defined populations, either to confer proof of concept in early development, or actually as orphan strategies all the way through to the market. We see many genetic diseases now in neuroscience that lend themselves very, very well to very selective precision medicine strategies. We're taking on 2 of these disorders. One is known as KCNQ developmental epilepsy. The other is a sodium channel developmental epilepsy. And we have very selective modulators that are going into one is in phase 3 and one is in phase 2 with Neurogen. And we will be looking to take these products all the way in these orphan indications. But these should give us a much higher likelihood of success, given the drug we have is really designed and developed for the mutation and for the variation that is really underlying the disease. I think another very important theme for consideration, at least as we've started, over the last half a dozen years or so, started to think about the commercial outlook, is really what are those drivers? It's not just about how well the drug works. The ease of use drug characteristics are hugely important. And of course, choice of endpoint, very, very important from a pricing standpoint, particularly in jurisdictions like Europe, where seizure reduction is probably not sufficient. One wants to try and identify in certain orphan indications, for example, developmental outcomes to allow premium pricing for a product as well. And that really, I think, is how it should be. We're also very interested in some of the non-small molecule modalities. We're starting to see a lot more interest in the antisense oligonucleotide and gene therapy treatments for neurological diseases. They're really not prime time yet, but there've been some successes. Spinraza, a nice example. Biogen has been a leader in the space, and I think we'll see a lot more coming out of the ASO oligo space, particularly in probably gene therapy and CRISPR space in the years to come. So these are just some of the key themes that we've started to think about at Xenon, and I think will become important elements, certainly as we look at neuroscience in the years ahead. So Kyle, I think that was really it. I'm happy to take any questions if there are, but I'll stop sharing my slide deck and hand over to you.
Great. Thanks so much, Simon, for that, for your overview of everything going on at Xenon, and for your insight into the CNS themes that you're seeing. So much appreciated.
Not at all.
Okay, I don't see any questions, but feel free if anything comes up to use the chat function. So next up we have Paul. Paul Brennan is the president and CEO of NervGen. He has over 30 years of experience in the pharmaceutical and biotech industries in Canada, Sweden, the UK, and the US. Mr. Brennan's previous employment includes executive roles in business development at various biotech companies, including Equinox, Arbutus, Aspriva, and Anormed. Mr. Brennan also held senior roles in global licensing and regulatory affairs at AstraZeneca. Mr. Brennan has contributed to over 10 products that have moved from development stage to approval and has a comprehensive list of business development transactions totaling over $3 billion in realized value, including the merger of Tecmira and Encore to create Arbutus, the sale of Espriva to Vifor for $915 million, and the sale of Anormed to Genzyme for $580 million. With that, I'll pass it over to Paul. Welcome, Paul.
Thank you. And just confirm you can hear me and
Yep.
see the presentation. Great. So thanks. And I'm going to do the same thing as Simon, talk a little bit about the company, and then also talk about my observations on the CNS space. And from my perspective, it's really interesting to enter CNS. I've been at NervGen now for 2 years. And prior to that, I've been in a number of different therapeutic areas: respiratory, cardiovascular, respiratory, infectious diseases. And so it's kind of interesting to transition to CNS and see how CNS differs. I used to think that oncology was really different than the rest of everything else in pharmaceutical development. Basically, there was oncology and then the rest. And now as I'm working into CNS, I really do see CNS as quite unique in many different factors, and it really does differentiate itself from other indications. So now I look at oncology, CNS, and then the rest. Okay, I'm gonna have some forward-looking statements in this presentation. If you have any questions about those, please refer to our SEDAR filings. So quickly, what's NervGen? Well, NervGen is a publicly listed company. We're on the TSX Venture, and we're clinical stage. We're developing a new class of neurorestorative drugs that repair damage to the nervous system. Now, neurorestorative is really key because most drugs that are in development for neurodegenerative diseases or for injury are trying to stop the progression of the disease or stop contained injury. And we're quite different. What we're trying to do is damage the repair that's occurred. And what this does is it allows us to do things that other products won't do. It allows us to see a response much more quicker and hopefully a response in fewer patients. Our technology is based on a class of molecules that are upregulated in the CNS in response to nervous system damage, and that inhibit the body's ability to repair the damage. So you'll know that if you cut your hand, the skin will repair itself. If you cut a peripheral nerve, it has an ability to repair itself. But if you damage neural cells, nerves, and glial cells in the brain, the nerves will not repair themselves. The nerves that die are gone forever. And what we've discovered is one of those components that play a key role in stopping that repair. What's really unique about our technology is that it has multiple modes of repair, multiple pharmacodynamic actions of repair. So typically most products that you see in development are doing just one pharmacodynamic thing. They're looking at lowering blood pressure or lowering lipids. What we're seeing is multiple modes or multiple pharmaceutical mechanisms of repair, including neuron regeneration, remyelination, and enhanced plasticity. The animal models that we have show quite spectacular results, and I'll show a video. We're well known for showing videos, and I'll show that in this presentation of the type of recovery that we're seeing. And this type of recovery is not typically seen in animal models. We're a phase 1 company. Our lead product is called MVG-291, and we plan to do phase 2 studies starting in 2022 in Alzheimer's disease, multiple sclerosis, and spinal cord injury. Okay, so I talked about how our technology is based around identifying a component that is responsible for inhibiting nerve repair. That component is a class of molecules called chondroitin sulfate proteoglycans, or CSPGs. CSPGs are a macromolecule. They have a core protein and a sugar side chain. They're present in healthy tissue as a structural component. But what happens is both inside the brain and outside the brain. But what happens is when you have injury, you've got a substantial upregulation of the CSPGs. And you get that upregulation both at the site of the injury and then throughout the entire brain. Those CSPGs are what's putting the brakes on nerve repair. And what they do is they'll inhibit the ability of the nerves, the axons to regenerate. They'll actually stick the nerves at the site where the CSPGs are. So if you think about nerves in a glial scar that forms at the site of a spinal cord injury, the nerves that are in the scar will get stuck. They'll become dystrophic. and they're there for the rest of the patient's life in a dystrophic state. They have other effects on nervous tissue. They inhibit microglial cells, or they create an inflammatory phenotype in microglial cells, and they inhibit autophagy. Now, we know these CSPGs are not just something that you see in animal models. CSPGs are present in humans, and they're upregulated as a pathology of disease. So they're seen in humans to be upregulated in spinal cord injury. They're in the lesions seen in multiple sclerosis. They're seen in patients in Alzheimer's disease. They surround the amyloid plaques. They're upregulated significantly in stroke patients and Parkinson's patients, and they've been seen in peripheral nerve injury. The next step in terms of the development of our portfolio was to identify the receptor by which the CSPGs were modulating their activity. That receptor is protein tyrosine phosphatase sigma, or PTP sigma. That was a really key step. That was identified in 2009, and it allowed our founder, Dr. Jerry Silver, to identify a path for developing a drug that would inhibit the activity of CSPGs. That drug is NVG291. It's a potent modulator of PTP sigma. And what we've seen in animal studies is that by modulating PTP sigma, we can promote axon regeneration, we can promote plasticity quite profoundly, and we can promote remyelination, as well as other repair mechanisms. Of course, what's really important is that those pharmacodynamic activities translate through to functional activities. And this is where we're really excited about our technology. What we've seen in 6 different disease models, I've only shown 5 here because one is outside the CNS space, we've seen functional improvement. And what we're seeing is improvement in motor control, both fine and gross motor control. And that's important because there are different neurological tracts that are involved in fine motor control and gross motor control. We're seeing improvements in sensory function, including detection of heat and cold, pain, and even something as complex as vision in optic neuritis models. We've got improvement in autonomic function, bladder control, and cardiac pacemaking in our AMI model. And then finally, we actually see improvements in cognition in our stroke model, measured by object recognition and ability to memorize. So I talked about our results being novel in our videos. This is a classic example of the type of a response we're seeing. What we see here is a spinal cord injury given to rats, and then they're treated with our product or placebo for 7 weeks, once a day, subcutaneous injection. On the left, you have an animal that's treated with placebo. On the right, you have an animal treated with our drug. And hopefully what you can see here is the animal treated with placebo actually has some limited movement with the legs, but no ability to coordinate, no ability to bear weight, and the tail is dysfunctional. On the right, you can see the animal has just a completely different level of functionality. This level of control, this level of recovery is unprecedented. In the experiment, the pivotal experiment where this comes from, Brink et al. 2018, the magnitude of response we're seeing is much larger than previously seen, and the number of rats that are responding to the treatment is much larger than previously seen. This gets us really excited. Now, that repair, those models we've seen not just in spinal cord injury, as I mentioned before, we've got models of multiple sclerosis, we've got models in peripheral nerve injury and other indications. We know that these CSPGs are upregulated anytime there's nerve damage, whether it's as a result of an acute nerve injury or an acute disease, such as spinal cord injury, stroke, traumatic brain injury, or neurodegenerative disease, such as multiple sclerosis and Alzheimer's. As I mentioned, we know that those CSPGs are present in the plaques in multiple sclerosis, in the lesions in multiple sclerosis, and they're present in the plaques in Alzheimer's disease. And these CSPGs are preventing the nervous system from repairing itself. Our goal is to take this forward, first in phase 1, healthy volunteer studies, and then in studies in patients. And so what we're planning to do is complete the phase 1 program by mid-2022 and proceed to patient studies in late 2022 in spinal cord injury, Alzheimer's, and multiple sclerosis. We're in the process of planning those studies with our advisory boards at the moment, and really excited about what those potentially can deliver in terms of results. Our phase 1 is progressing really well. Currently, we've completed the single ascending dose portion of our study. What we've reported so far is that the side effects are mild and transient, no effects on vital signs, ECGs, or laboratory assessments. And the doses that we've achieved at the highest SAD dose cohort are already 100% higher than the equivalent highest dose in our efficacy studies, and over 100 times higher than the lowest efficacious dose seen in our animal studies. Also, our PK looks really good. The drug is rapidly distributed. The half-life appears to be longer in humans than in animals, which is a positive sign. And so with that, I'll finish talking about NervGen and talk a little bit about my observations in the CNS. And I think first, what I always like to do, based this is really my large pharma background, is to look at where are the diseases that most people are going. And really, the diseases where there's the most activity are neurodegenerative diseases, which is really the #1 focus, psychiatric diseases, pain, autism, stroke, epilepsy, and then not a disease, but psychedelics is now taking on an important component in the CNS space. Another observation is that CNS has typically been seen as a very difficult and risky area of product development. And you can see this is, I think most people are familiar with the clinical development success rates that were published by BioTracker in 2016. This is a little bit old now, but you can see in neurology and psychiatry, the success rates have typically are lower than all indications. And that's one of the reasons why it's been seen as difficult and risky. However, I think as Simon mentioned, CNS is now seen as the new oncology, or as oncology was 20 to 30 years ago. If you go back to oncology 20 to 30 years ago, you were mostly talking about chemotherapies. And then you had the advent of targeted monoclonal antibodies, which changed the space. We're seeing the same thing now in the CNS space, where people are saying, as the advent in genetics is becoming more sophisticated in CNS, following on, as you see the continued success in the MS space, and the perceived success in the Alzheimer's space. And I'd also like to really add to that the success that Xenon had recently in epilepsy. There's a belief now that CNS is about to break through. And that's really exciting for early stage companies in terms of our ability to finance. And then finally, the last observation, it's really interesting to see Alzheimer's disease and what's happening in the Alzheimer's space. It is the wild west of pharmaceutical development for many reasons. First, there's a lot of unknowns, and it's in no way fully understand what is the cause of Alzheimer's disease. There's definitely a herd mentality in Alzheimer's, specifically in the amyloid beta and tau space. There's been research in that area for over 20 years, and only one product approved in the space, and it's marginally approved. And still, amyloid beta and tau make up 1/3 of the development pipeline. There is incredible reward for those that succeed in the Alzheimer's space. The market potential, 6 million patients, expected price of a new effective therapy, about $50,000 a year, gives it a market potential of about $300 billion. The first effective drugs are widely expected to be the largest selling drugs in the world. The pharma upfronts for preclinical projects are crazy. They're over $100 million in total deal size, with $1 billion for preclinical projects in the Alzheimer's space. And then companies leading with Alzheimer's drugs have had really quite substantial valuations with very little data. There's 2 companies that have had over $1 billion, even $2 billion valuations based on data in less than 10 Alzheimer's patients treated for less than 28 days. And then finally, just like the Wild West, there are bad actors and fraud in space. In the past 6 months, multiple AD companies have been accused of fraud and forgery and lost significant market cap as a result. So you have to pay attention and watch out. Make sure that you do your diligence when looking at Alzheimer's companies. And with that, I will end my presentation. I'll see if there's any.
Yeah, I think you have a couple questions there, Paul. Can you
Sure.
Can you see them?
Yeah, so I'm in the chat. I'll go into the Q&A. So one's for Xenon, one's for me. And the first one, have we investigated psychedelics as a potential treatment for specific indications? No, we haven't. For us specifically, we have our hands full with our mechanism. There's so many different indications where we can take our technology that going into psychedelics really isn't within our purview at this point. Are the changes seen in the drug candidates permanent, or does the continued drug application need to be carried out in the effect? That's a great question. What we see is we've studied in the rats, those animals for up to 7 weeks. We demonstrate for 7 weeks and we follow the animal for 7 weeks. And what we see actually is not only after we stop administering our drug, we actually continue to see an improvement in the animals after that administration for 2 to 3 to 4 weeks after the administration, and then it plateaus. We've never seen a significant reversal of the gain that we see. And really that's a function of what we're doing is we're allowing the animal to set new connections, to ignore the CSPGs and create new connections, either or remyelinate. And once that new connection is able to form or the remyelination is able to occur, you don't see a reversal of those connections. At least we haven't seen through the functional response.
Great. Looks like that's it for questions. And in the interest of keeping things moving, I think I'll introduce our next speaker. Thanks so much, Paul. Very interesting stuff.
Thanks very much.
Okay. So next up, we have Michael Mee. Michael Mee is a principal with Amplitude Ventures, a precision medicine-focused venture capital fund that creates, builds, and grows companies. Following his PhD research in biomedical engineering in George Church's lab at HMS, Michael has been working as an associate at Flagship Pioneering since 2015. His time at Flagship has been focused on the development of 2 microbiome companies in the agricultural and therapeutics domains, co-founding and launching a new therapeutic delivery platform and gene therapy company, Sana Biotechnology, and most recently developing new ventures in AI-based drug development and gene editing areas. A long-term goal of his has always been to bring learnings from Boston back home to help contribute to the Canadian biotech ecosystem. To that end, he has recently relocated to Montreal and is excited to be able to achieve that goal along with the growing team at Amplitude. Thank you, Michael, for joining us.
Thank you, Ryan. Thank you to the organizers, and thank you to all the attendees that are joining us this afternoon. Just getting things sharing here. Awesome. And while I'm at that, I was told I should be sort of sending links through the chat, so I just did. It's easy to find via Google anyways, but there's the Amplitude website, and would draw anyone interested their attention to the actual careers tab that we have there that highlights a number of really exciting opportunities at all levels of the organizations across our portfolio for you to have a look at. Awesome. So we'll try to move fairly quickly, but still reserve some time at the end to address any questions that come through the chat. So similar to the other speakers today, wanted to start by providing a bit of context and explanation about Amplitude and who we are as a fund and what we do. So Amplitude is one of Canada's leading healthcare investing platforms. We have a $200 million fund, which is really the largest debut healthcare venture fund in Canadian history. Our investment strategy looks to capture the full innovation value of intellectual property generated by Canada's world-class academic institutes. And we have sort of a deep bench across 3 of the main innovation hotbeds in the life science industry. Vancouver, Toronto, Montreal, our team of 10 is distributed across those. And as was highlighted in my introduction, we really focus at investing across the various stages of development of companies, everything from company creation to sort of building and growth of those opportunities as well. And I'll go into a little more detail on that and focus on what I think is one of the more unique elements of Amplitude in our company creation engine. So how do we do this? Where does this money come from? We're supported by some of the leading institutional investors across the country, large pension plans, institutional investors, corporates, and very grateful for their support and interest in supporting what is really a burgeoning and re-burgeoning sector within our economy. And critically, what we also look to do is not just invest our own capital, but really leverage the capital that we bring to the table and invest in companies by bringing around the table leading international co-investors. to direct a large amount of leading foreign investment into Canadian life science innovation companies. So this really supports the acceleration and sort of full-on support of these companies to drive to significant IPOs on sort of international markets. So in terms of our strategy, sort of everything starts from identifying really exciting competitive innovation emerging from our centers of excellence across the country, investing in these companies to provide that acceleration capital that can drive to significant value creation in the first 5 to 7 years of that company's development. And critically, building out world-class management teams by bringing around sort of global talent players in the life science industry to help scale these companies. And really scaling that access to talent and capital by sort of focusing our investment in these clusters and areas that we think we have a competitive advantage and sort of really rich technology across the country in the medical sector is in cellular and targeted therapies. We're seeing a sort of really robust cluster of that developing in Vancouver with a number of the major companies that are based there. And then sort of our sort of world leading capabilities in AI and applied machine learning that we're seeing applied and deployed into next generation imaging and intelligent devices, but also small molecule and targeted therapies as well. And so really exciting developments there. So we're highlighted create, build, and grow as elements of our strategy. This is just a snapshot of the portfolio as it stands right now at the various stages that these companies are in. So our create, and I mentioned I'll go into this a little bit more detail, but looking at the formation of companies, bringing around very experienced management teams, CEOs with sort of the biggest ideas that we can identify. And we have a few different models in terms of how we approach that I'll go into in some detail. We'll traditionally sort of seed those with anything from $1 to $5 million to kick things off, but then continue on with further investments up to $20 million to support the growth of that company. For our build category, these are generally Series A or Series B investments, where we'll be coming in with an initial check size in the $2 to $8 million range, follow-on investments increase beyond that, where we're looking to scale the management, fill gaps in the team, and really elevate the company's strategy to grow new options, further expand the pipeline of therapies under development, and really provide that capital for continued platform development of those companies. And then lastly, our grow investments, which are the investments in companies at the latest stage of their development, including companies like Repair, Deep Genomics, and Prolenia in the neurospace, which is the topic of conversation today. But these are investments, again, that will be crossover investments, mezzanine investments prior to going public. Our initial investments in these companies will range sort of in the $10 to $20 million window, and looking to sort of push to clinical milestones in these companies. So wanted to provide a little bit of a perspective of where our view on sort of the current condition of the Canadian life science ecosystem. And I think we're in a great position right now. The Canadian life science innovation sector is really in a period of renaissance, with issues of the past being overcome. So some of those in our perspective are, previously there were too many companies getting too little sort of amounts of financing that really led to this trickle of financing approach that is very hard for management teams to deal with and really make significant innovation and drive to significant milestones. And so just some figures to sort of look at here. Back in 2016, the average deal size in Canada was about 2.5 million. Today, the average deal size is 22.1 million. So a significant sort of step up in amount of capital that we're providing these entrepreneurial teams to drive and create value in their companies. More and more, we're seeing companies being created with really exciting platforms. I think both Simon and Paul spoke to that element of the ability of companies and technologies to produce multiple therapies. These are really the types of companies that are sort of capturing the imagination of investors and sort of seeing the possibility for being competitive on a global stage. And so this attracts a lot of international capital and critically attracts a lot of talent into these organizations as well. And then maybe most interestingly, we're seeing a sort of refocusing of domestic capital and the domestic capital base in institutional investors across the country into this sector. And so in the table on the bottom right here, we're seeing sort of the amount of capital going in for life science investments coming from Canadian institutions, Canadian investors, more or less being on par this year with American investors, which is a very positive signal for continued sort of support and development of these capabilities within our borders. So now maybe focusing a little bit more on neurological diseases, echoing a lot of the sentiments and messages that both Simon and Paul mentioned earlier, but wanted to break this down in terms of what we're seeing as opportunities and emerging trends, and talk a little bit about the emerging and enabling infrastructure that we have across the country to support innovation in this sector. So as was mentioned previously, this area remains sort of one with significant unmet need that is attracting more and more investment. Sort of echo those sentiments that we're seeing echoes of what oncology and sort of innovation in oncology looked like in the decades of the past, sort of being applied here to neurological disorders. So really seeing more investment to develop these novel science, novel targets and insights around targets and new and advanced therapies in neurological disorders. Critically, a lot of these disorders are sort of fundamentally heterogeneous etiologies and can be sort of driven by a number of underlying mutations and sort of underlying conditions. And we think a critical part of really unlocking therapeutic development here is going to be the development and advancement of novel biomarker strategies to really enable precision medicine, improve patient stratification and dose selection. And these range from genetic computational approaches, protein approaches, digital biomarkers to sort of brain mapping approaches. So I think there's a plethora of things in development that are really exciting and will enable sort of the next generation of therapies in this space. And then additionally, we're poised to sort of really take advantage and better understand this biology with the emergence over the last decade of really leading high resolution technologies to expand our mechanistic understanding of these diseases. And sort of expanding our view and understanding of drug targets and cells that are relevant in these diseases beyond neurons. I like to call it the non-neuronal view of disease in this space as well. And finally, we have strong fundamental infrastructure in this regard across the country, from internationally recognized leading academics in this field to build that strong base of human capital and intellectual property, and leading precision cell targeted therapy, stem cell and organoid capabilities that we'll hear a little bit more with the next two speakers to drive development of novel therapies in this space. So as I mentioned, I want to spend a little bit of time talking about Amplitude's company creation model, because I do believe it's a little bit unique and maybe something interesting to focus on in the context of this breakfast speaker series. But we have a few different approaches. And so we think of this as sort of in this table, the ability to in-license into a company that we created, leading IP from one of the world leading academics across the country. So an example of this is Radiant Biotherapeutics within our platform, which is a multimeric binder biologic therapy platform that we're developing. Sort of working with founding management with initial concepts to really expand the strategy and expand that management team to unlock the capability of the technology. Working deeply with academics who want to take a leave from their academic position and take the helm as CEO. So we've done that a number of times as well. And then sort of lastly, in the final column, thinking about our ability to leverage our internal company creation engine that is stemming from our fellowship program to generate new companies. And so I'll speak in a little more detail to that point right now. So this is something that we launched last year. We brought around the table 6 leading innovative entrepreneurial scientists from across the country to work with us in an intensive program for 3 months over the summer. The focus of this is really to drive an evolutionary process of company creation, starting from really interesting insights emerging from academia, from publications, some that may be dogma challenging, but really pique our imagination, and pushing those forward to understand how they can be evolved and developed into a broad therapeutic platform with a strategy behind that identifies how to sort of unlock pools of value in different therapeutic areas and diseases. In doing so, we're really seeking out what we call white space. areas of technology and innovation potential that aren't very well trodden, and have the opportunity to lay down pretty broad foundational IP to support the development of these companies. And then we take the output that comes from the development of these venture hypotheses, and following that, we'll sort of continue to work with these fellows sort of for the rest of the year on further developing these concepts and refining these in what we call a prototype company, really testing this and putting this into action, at which point when we feel everything is looking good, we'll inject a significant amount of capital and continue to build that company and recruit around a management team. In all these early stage company creation events, time is of the essence, and wanted to provide a little bit of perspective from Repair. And critically, all of this is motivated by the last bullet on this slide, right? Where unique technology is only unique for a short window of time. And so we really need to think about how to move quickly. And it's really important that we have this mindset across the country that time is of the essence. So with Repair, this is a company that went from a seed round to an IPO in 4 years, with the Series A in 2017 to an IPO in 2020, and significant value creation during that time, where sort of starting from a pre-money valuation in the initial round of around $12 million to a $750 million pre-money valuation at the time of IPO. All of this driven by significant platform development that really enabled a very, very broad pipeline of therapies in development. And this was built in collaboration with leading academics from around the world, drawing multiple different insights, multiple different pieces of IP to pull together. And then additionally, there are elements that are sort of accelerating programs. And so there was a very interesting program in development at NYU that was able to be leveraged to really accelerate the chemical matter that the company was working with, to have a program that was very far along and ready to move into the clinic in a pretty short time. So both the breadth and the depth and the speed of that lead program are important for companies to create value in an accelerated manner. Wanted to say a little bit for those emerging entrepreneurs, academics in the audience, to think a little bit about what we're looking for at Amplitude and the ideas that excite us the most. Of course, we need very strong foundational science, really thinking about next generation technologies, things that have the potential to become the next generation standard of care. We sort of really look for the ability to think critically about what are the gaps in management and how to fill those as quickly and effectively as possible. We like to engage with entrepreneurs to understand how quickly can we raise capital to grow the organization very quickly through both organic and inorganic approaches to drive that value creation. And then lastly, that sort of initial vision and ambition of being a global champion. And I've too often sort of spoken to companies where I've heard that this is going to be the best company in Canada, and we really need to be thinking about competing on the global stage in this international industry. So clear storytelling, leading with the most exciting and interesting data, thinking through the long-term vision of the company, and not just where you're going to get in the next year, but what does the company look like in 5-7 years from now? How established will it be? And sort of a clear articulation of those upcoming milestones and timelines to achieve that value creation are all things that we'll be looking for and engaging with the entrepreneurs on. So with that, I'll conclude. If there are any questions, comments, happy to engage right now or offline as well. Always happy to connect.
Okay, great. Certainly, if anyone has any questions, there's the chat function. So thank you so much, Michael.
Awesome.
Okay, in interest of keeping things moving along, we have 2 more speakers. So let's move on to our next speaker, Eric Roos. As Chief Business Development Officer, Eric leads the expansion of Aspect's global partnerships with biopharmaceutical innovators. He has over 30 years of experience in the development of biopharmaceuticals, cell therapies, and regenerative medicine products. His leadership roles in the cell therapy industry spanned a multitude of responsibilities, including leading early phase development, process optimization, clinical trial execution, and business development. Most recently, during his decade-long tenure at Thermo Fisher Scientific, he was responsible for building global strategic alliances and expanding their cell and gene therapeutic business unit. Welcome, Eric.
Well, great. Thanks, Kyle. Great to be here and be able to present where we're going with Aspect amongst this esteemed group of presenters. It's very exciting times for Canada, and certainly the neural space is interesting. We're going to take a little bit of a different track as I get this deck queued up and focus more in endocrinology at this point, given our technology base, which is really looking at taking bioprinting and moving technologies forward in that space. So let me get to the front here, and we'll start here. Thank you very much again. So let me jump into it. I think, what we're doing here at Aspect is very, very interesting from our perspective. And I think we're really opening the doors to a new class of medicines, through cell therapies. But what we're doing is taking a platform technology, combining that with therapeutic cells and biomaterials, and we're looking to create bioprinted cell therapies. So exactly, what are these? These are therapies that can be implanted and execute function for organ systems and tissue damage, and repair those systems and provide benefits for patients from that perspective. So we recently did a Series A round in the beginning of 2020. We've grown the team to about 60 folks here in Vancouver, Canada. We have a very strong patent portfolio revolving around our microfluidic platform, and I'll talk about that in a minute. We've been applying that technology to a number of different areas. Some have been more model-based, where we've looked at creating systems where drugs can be screened. But we've really pivoted and are focusing now on therapeutic development and the build out of our internal pipeline. We're also encouraging external innovation through a discovery ecosystem. And that really allows us to, as we're building out our translational capabilities here within Aspect, we can take novel technologies and bring them through our systems and get into the clinic with those novel technologies that can apply our platform tech here. We're really focused on developing strategic partnerships, as mentioned in the intro, and are building relations with companies at the present time to help us take our early phase programs into the clinic. So to go a little bit deeper into what we're focusing on specifically in these verticals, we're looking at three verticals. One is really an encapsulated cell therapy, where we're printing tissues, so cells from various organ systems that have a certain responsive nature to them, so they can sense a body's deficiency. So for example, when we're encapsulating pancreatic tissue, we can sense glucose levels, and those pancreatic tissues can secrete insulin in relation to the requirement. So this is really kind of a replacement of lost function and encapsulation perspective. The second vertical that we're looking at is really more focused on true tissue regeneration. So these are integrative technologies where we might create and print tissues that are introduced into the body and repair a damaged tissue. This could be in ligaments at some point, vasculature, potentially peripheral nerve injury as well. And these are concepts that our platform can certainly be applied to, and we're looking at ways we can execute on those. And then the third vertical is really taking engineered cells that have been engineered to put out either proteins or antibodies that we can then encapsulate, essentially create a bioreactor for a steady state delivery of a therapeutic modality or compound. So that's those are the different areas that we see our platform being able to make a difference and impact patients' lives. To go really quickly through our technology, we have developed this over the years, where we're really the technology space here in the microfluidic printhead. And it's that printhead that allows us to take multiple channels of materials at very measured rates in a very easy to the cell format that allows us to cross-link them and encapsulate them at the same time within various biopolymers. And with this printhead and the control of this instrumentation, we can create multiple types of structures and architectures that you can see here, really based on a fiber type of an architecture and format. And then we take that fiber on a micro scale and then pattern that to create 3D systems that will have the appropriate extracellular matrix, et cetera, that maintain cells and let them execute their metabolic function or repair characteristics. So a quick little video here to give a little bit more insight into what happens in that microfluidic chip. We can control and introduce multiple materials and bioinks that will, again, maintain the environment of the cell, and we can optimize that environment so that they are as responsive as it can be. We have a lot of levers that we can pull to create various shell and core structures that, again, can be used to optimize cell viability. The switching between various channels is very easy to do for this particular system. And then we can then pattern these devices to make sure that we have appropriate numbers of cells to affect therapeutic outcome. So our current focus is really in type 1 diabetes and pancreatic tissue, as I touched on. We are also getting some very interesting results in the liver space. So the printing of hepatocytes to potentially be applied in various liver diseases, including acute liver failure, but also rare genetic disorders. And we have multiple discovery programs, as mentioned as well. So we're really looking to and are building out our translational capabilities to execute and move towards clinical trials here. So now to go into a little bit more detail in what we're doing with this bioprinting, you can see some results here of the microscale, small scale structure where we're encapsulating pancreatic tissue, implanting it and demonstrating that we have a very high viability post-explant of these tissues, as well in the case here of pancreatic tissue, a very high staining, dithazone staining for insulin, again, post-explant. So indicating that we can encapsulate these cells, place them in vivo, pull them out and have them very highly viable after that explant, as well as being functionally active. So we took that concept and went into mouse studies in a model of diabetes, where we would treat a mouse with streptozotocin, an implant on day 0. So what we can see here is a spike in blood glucose, and then at the point of implantation, glucose drops significantly and is maintained over 80 days. So we have a very active and viable, responsive, encapsulated cell system here that can maintain glucose homeostasis. We see that we have a very uniform output of C-peptide, which is a correlate biomarker to insulin. So that control is maintained over the course. These are random measurements, so there's no control over whether the animal is eating or not at the time of measurement. So there's some variation there, but broadly speaking, it's very steady. And in the third graph on the right, we have been able to show that the kinetics over a 3-hour period to as experienced with an oral glucose tolerance test is very similar to those mice that had their pancreas knocked out and had the implant placed in their peritoneal cavity to controls. So we were demonstrating a very physiologic response to our bioprinted systems. So that is wonderful when we can do it at a small scale, but the real trick is getting that scale to be increased so that we can start looking at evaluations in humans and execution of clinical trials. So we took our script and the technology behind our platform to create a structure. As you can see here, we've been able to encapsulate the beta cells or pancreatic islet tissue with very high viability and can also demonstrate glucose responsiveness in vitro with these 3-dimensional pattern systems. So wonderful to do that in vitro, but the real trick is, can these devices work as they did in the smaller scale devices? And the answer is yes. So we've been able to implant these now in a larger model. So these is a rat model. So the same essential system there where we would introduce streptozotocin, knock out the pancreatic function, and then following implantation, we see a return to glucose homeostasis. So we're very encouraged by these results. We now have a system that we can print using our platform technology. We can demonstrate that it's working in a larger animal, and we're at the point of discussing how best to proceed through an interact meeting with FDA and getting ready for larger animal studies and an eventual IND application. So from a platform perspective, that's wonderful, but what else can we do? We've been looking at hepatic encapsulation, as mentioned previously as well, and we're seeing very similar types of functionality that can be maintained. So again, maintaining hepatocytes, encapsulated hepatocytes over a period of time. In this case, it's been 28 days where we are able to demonstrate metabolic output as measured by albumin in this middle graph, and then post-explant demonstrate, again, albumin release so that we can demonstrate these hepatocyte encapsulated implants are still functional post-explant, but more interestingly, we can see that we have ammonia detoxification, which is very, very interesting because that ability for these cells to detoxify ammonia really lends itself to the opportunity for being a bridge to transplant for patients that undergo acute liver failure. So that's a very exciting piece of information and really encourages us from the expansion of this platform to other modalities as well that we touched on earlier. We're also seeing some very interesting maturation. I mentioned at the beginning that we can control the environment that these cells are placed in. We can create organotypic types cultures that really mature in a very unique way once they are in vivo. And what we're seeing here is essentially an image of hepatocytes that have organized and polarized themselves to create bile ducts. And that's quite interesting from the perspective of the potential for applying these type of constructs to rare liver diseases where enzyme deficiencies might be present in a disease state or a diseased individual, but the ability to then secrete enzymes that are naturally produced by these hepatocytes to affect that rare liver disease is something that we're really looking to drive forward as well. And we have some early indications of this where we're looking at a specific enzyme level across multiple animals and show that we can actually detect in a serum enzymes that are representative of rare liver diseases. So very exciting opportunity there. So I guess just kind of in summary here, what we're doing again is taking this platform technology and driving towards therapeutic applications and taking that platform and encapsulating cells at this point. We have multiple verticals that we can go after. We feel there are a lot of opportunities to take this technology forward, but we're really focusing very much at the moment on pancreatic type 1 diabetes and liver disease. And of course, none of this could be possible without a fantastic team. And we've been very fortunate to get some wonderful people, scientists, and developing the leadership here at Aspect to drive this technology forward. And we're firing on all cylinders and really excited about future interactions with FDA and moving into the clinic. So I'll stop there and happy to take questions if there are any. And thanks again for letting us provide an update to you.
Great, thanks so much, Eric. Again, if anyone has any questions, the chat function is available. So thanks, Eric. I'll introduce now our next speaker, who is Dr. Stephanie Willerth, CEO of Axolotl Biosciences. So Dr. Willerth, full professor in biomedical engineering, holds a Canada Research Chair in biomedical engineering at the University of Victoria, where she has dual appointments in the Department of Mechanical Engineering and the Division of Medical Sciences. She also holds an appointment with the School of Biomedical Engineering at the University of British Columbia. She serves as the acting director of the Center for Biomedical Research and the Biomedical Engineering undergraduate program at the University of Victoria. She is an active member of the steering committee of the BC Regenerative Medicine Initiative and the Stem Cell Network. She also serves as a staff scientist at Creative Destruction Lab. She was the president of the Canadian Biomaterials Society, serving a 3-year term as president-elect, then president, and past president from 2017 to 2019. She recently founded the startup company Axolotl Biosciences. Welcome, Stephanie.
Yeah, thanks so much. It's an honor to be here today, and let me see if I can get my slides going. Thanks again to Life Sciences BC. And one quick note is I did actually manage to get out of all my administrative roles. So currently my major focus has been on running Axolotl this semester, since those had come to an end. And also just another shout out to Life Sciences BC, because we did graduate from their investor readiness program this past spring. So we founded Axolotl. It was spun out of my research lab at the University of Victoria in March 2020, coincidentally the week of the pandemic. So it was definitely a time, but I've got a lot of really exciting stuff to talk about today. And building upon what Eric had talked about with Aspect, I really do think 3D bioprinting has the potential to transform how we study a lot of these diseases and disorders, especially of the nervous system. So overall, the mission here at Axolotl Biosciences is to deliver superior quality reagents for printing human tissue models, which make research and drug discovery easier than ever. And while we saw Aspect's technology, and we were actually my research lab at the University of Victoria, we received the second one of their systems, and we've actually published a lot of work using their system to bioprint neural tissue models, which I'll talk about in a little bit. What our company is really focused on is not making a printer or making therapeutics, it's actually making the tools, and in this case, specifically a bioink that lets you print these stem cells at high levels of viability and function. And that's really where our focus has been. So we are essentially a reagents company, although we have partnered with several other companies as well to go towards some of the clinical applications as well. And so I know Eric didn't get into this much, but when you bioprint, it's similar to traditional 3D printing, except instead of using plastic filaments, you're using specialized bioinks. And one of the big challenges in the field has been getting inks that would actually allow stem cells, and particularly pluripotent stem cells, to both survive and differentiate into functional tissues that work like they do in your body. And he did present some really lovely data from their recent studies and other applications for that. My research group at the University of Victoria and throughout my career has been heavily focused on neurological diseases and disorders. And so we had spent a lot of time optimizing a certain protein in order to get 3-dimensional functional neural cell cultures. Just to give you an idea for some of the market segments for why we've chosen to sell bioinks, our initial target audience and who we actually currently sell to are academic researchers. And this is around a $590 million market. You can see the second largest group is pharmaceutical companies, and we have been looking into partnerships with them, then followed by cosmetic companies and clinicians. But this is a rapidly growing field, and I really think that things like our bioinks that we have that have now enabled the printing of stem cell-derived tissues really can transform bioprinting as a tool for personalized medicine. And I think this is really important in the field of neurological diseases. And I know, as one of the previous presenters mentioned, they were talking about oncology and how everyone thought oncology was different for years. But I think we're going to actually start to see that now as it becomes easier to print some of these human tissue models of neural diseases, that things like Alzheimer's and Parkinson's are going to start to see, similar to cancers, certain types of these diseases are going to respond better to treatments than others. And we're going to really start looking at personalizing medicine for neurodegenerative disorders, similar to how it's currently done for the different types of cancers as well. So our product, which is actually currently available for sale, is a novel versatile bioink called Tissue Print. And we've published this in, I think, 6 or 7 different journal articles now. Our original formulation, which is compatible with the Aspect system, is featured in the 2019 Methods of the Year issue of ACS Biomaterials and Science. Because at that time we were using it to print human-induced pluripotent stem cell-derived neural stem cells into bioprinted tissues with really high levels of viability, as well as the cells were expressing mature neuronal markers. So we were able to print neural tissues that both survived and also acted like the tissues you would find in your spinal cord and the brain. More recently, we did a full characterization of the properties of our bioink that came out in the journal Processes, and it was featured on the cover. We currently have 2 different versions of our ink for sale, the version that's compatible with the Aspect printers, and then we have another version that's compatible with cell ink bioprinters, as they have thousands of bioprinters installed worldwide. And so it's a bigger market in terms of who we can sell our products to. And so, as I mentioned, what separates our product from some of the other reagents and inks on the market currently available are you can print these delicate stem cells and actually get them to survive, but also function and signal like they do in the human brain and spinal cord. We've got a lot of different beta testers out there, including 7 companies, including CellInk and Brinter and RegMat, who all make 3D printers. Another one of our really interesting collaborations I won't get into today, but just to show sort of the potential of the bioinks with bioprosthetics, is they've used our ink in combination with some other technology to print functional human bone tissue, and they're further pursuing this partnership with us. We've got a number of academic beta testers who we've converted to customers, both here in Canada and in the United States as well. And we've currently recruited a cohort of second group of beta testers for our second bioink product as well. So our base ink, which again, we had developed in our lab as a tool specifically for printing neural tissue, actually has turned out to be quite versatile. It can print human vein endothelial cells, so things you'd need to print blood vessels. We printed pretty much every type of neural cell you can imagine, from neural progenitors to functional motor neurons in aggregate. I'm also going to show some really exciting data from a paper we had to come out last year, where we actually did a really complex human glioblastoma model, and we're able to use this as a tool for drug screening, which is where I think the real potential of bioprinting lies in. We've had some papers come out where we've printed mesenchymal stem cells. Our collaborators in Texas have used tissue print to print cardiac cells that functioned as well. And in collaboration with Ryan Flanagan's group over at the Vancouver Prostate Center, we've printed some functional testy organoids, and that's on bioRxiv if anyone's interested in learning more about that. So the thing I really wanted to just to show you the power of 3D printing is talk about a collaboration we have with a company based out of Montreal called Zonula, who read one of our papers where we had bioprinted a glioblastoma model, and they wanted us to use it to evaluate a potential drug target for treating glioblastoma. So in this case, they have an incadherin antagonist, and for those of you who might not have taken cell biology recently, incadherin is one of these cell adhesion proteins, which essentially helps the cancer cells stick together, and it's highly overexpressed in these cancers. And so the idea is this antagonist would be able to disrupt these compounds and in turn kill different types of cancer. And so they were curious to see how this would function in our 3D model of glioblastoma. So this was using the Aspect system. We did use our tissue print in order to print ring structures, where we actually printed these human glioblastoma cells alongside healthy human astrocytes. So this is a co-culture model where we had our diseased tissue alongside healthy tissue. And interestingly, from this study and previous studies, when you actually grow the human cancer cells in our ink, they form tumors just like they do in the brain, and they're really hard to break up, just like it is in the brain. And so this is, I think, a more representative model than using 2D culture. And while I won't get into in this talk, Imka, who did this work, actually showed a bunch of different controls as to why we actually needed the 3D bioprinting aspect to get the right ratio of healthy to diseased cells and actually get these phenotypes for drug screening. The other thing she did that was really interesting is she actually allowed these constructs to culture for 16 days, forming tumors before we added the compound. So this was simulating something more similar to what you would find in a cancer in vivo environment compared to just, say, culturing cancer cells in 2D and hitting them with your drug. The other thing she did with this was she labeled the glioblastoma cells with green fluorescent proteins. It was easy to track our tumor size and measure them, while the astrocytes were not labeled. And so here you can see we treated it with different doses of this drug, and at higher doses, you can pretty easily tell visually that the higher doses we are killing off these tumor cells while sparing the human astrocytes. So this was quite interesting for a number of reasons. The model's a good tool because it can tell you if your drug is just killing all your cells or just selectively killing your cancer cells. And then using the labeled cells also enables you to visually track these effects over time. And that's what Imka did. So if you want to read more, this came out in Biochemical and Biophysical Research Communications last year, but you can see significant decreases in the amount of tumor area at these later time points. And so, as I mentioned, while our company is a reagents company, this is where I think the power of bioprinting lies, is that you can actually make these fully human tissue models and assess drugs in ways that you can't. And a lot of the currently used cancer models involve putting a tumor into an immune-compromised mouse and then seeing how the drug reacts in terms of treating that tumor size, but you're missing all of the human immune components. So this system, well, in this case, we just had healthy tissue versus the cancer cells. You could also eventually envision a system where you had not just those cells present, but all those cells of the human immune system present. And so then you could see how your treatment works in the effect of a fully humanized system. And so I'm not going to talk about it today. One of our collaborations with Dr. Hakka Nygaard over at the University of British Columbia, where he runs the Alzheimer's clinic, we do similar work, except we use patient-derived HIPSC lines to bioprint our neural tissues. And we have characterized them, and you can track some of these Alzheimer's symptoms, including tau and amyloid beta, however controversial they may be, in our in vitro model. And as our tissues age, you actually see these increase. So we do have a way to track sort of the level of Alzheimer's present in these tissues. And so we're actually hoping to use those now as a tool for drug screening, because if we were to find something promising, potentially, because Dr. Hakka Nygaard is an MD PhD who does see patients, he could potentially prescribe the drugs we find back to his patients. And so that's a really cool way of sort of taking the whole process full circle. And going along with that, we recently received funding from Innovate BC to develop our second product at Axolotl called BrainPrint BioInk, which is based on our patent pending technology. And so we have we have these drug releasing particles we incorporate into the space BioInk of tissue print. And it really effectively promotes the differentiation of human induced pluripotent stem cells into functional brain tissue. And so we're currently in beta testing with our BrainPrint BioInk. We did publish a prototype, which came out in Frontiers in Biotechnology and Bioengineering last January, and it's already been cited 45 times since it came out. So there's huge level of interest in this. Along with our patent technology, this actually covers any type of bioinks that has these drug releasing compounds. So we also are currently developing a similar product called HeartPrint that will enable the printing of heart tissue. So if you know anyone who's interested in being a beta tester for either BrainPrint or HeartPrint, definitely feel free to reach out. Or if you're interested in learning more about using tissue print as well to print your own neurological models of diseases for drug screening applications. I would like to thank my team. Obviously, it's been a crazy ride since founding the company in 2020. My co-founders include Dr. Laura de la Vega, who really helped drive forward this technology, along with our other co-founder, Lila Abelseth. They are co-authors on several of the publications. So if you just Google Scholar my name, you can sort of read about all the work we've done in the neural field. We currently have a full-time team of 4 staff, along with several co-ops, and we just recently welcomed another intern, Sal, who've all been lovely to work with as we've been beta testing and now shipping out our product for commercial sales. And I think one of the things we do really well is help people troubleshoot when they are working with our products in order to actually get some really nice data coming out. And we do have a couple of publications with our beta testers that are in preparation for submission. So you'll start to see some more of our tissue print coming out in scientific journal articles as well. With that, thank you for listening. Please check us out on social media. We are currently fundraising. We are in talks with some of our major partners about doing some deals, but we are currently seeking investment. So if you're interested, feel free to reach out as well and check us out on social media. I'm also on LinkedIn and happy to connect. And thanks everyone for listening. And I guess I'll see what's in the chat.
Great. Well, thank you very much. And we'd like to thank all of our speakers, Dr. Simon Pimstone, Paul Brennan, Dr. Michael Mee, Eric Roos, and Dr. Stephanie Willerth. And thank you to our event supporter, Blades, and in particular, thank you to Kyle Misrich, partner, and Joe Garcia, partner as well. So we have a few events coming up on December the 14th. We have our showcase series in Prince George. The January 11th, we have our Gardner webinar. 21st, we have Career Connect Day presented by Biotalent Canada. The 27th, we have the McCarthy Spotlight speaking series, all on oncology. And February 16th and 17th is Access to Innovation 2022. Lastly, I'd like to thank all of our sponsors. We are very grateful for all of your continued support. And I want to thank you again for attending, and have a great rest of the day. Thank you very much, and take care.